So, welcome to my channel. This is Dr. Muthu, Assistant Professor, School of Sciences and Humanities, and Lesser University, Warangal. So, in this today's video, we are going to cover binding affinity prediction. So, how the protein, which is also known as macromolecules, and ligand, which is also known as small molecule, how these two are interacting together and bind together to form a complex structure. And what are the possible ways for the binding mechanism? What are the visualization softwares to evaluate or to visualize the bindings? And there are some softwares, free softwares and paid softwares where we can perform the binding affinity calculations. So all these things we are going to see in this today's lecture. Let me start with some of the theory part we will be covering in the first half of this video. Then we will move on to the practical part. Some of the servers available, so we can perform hands-on training session like sample protein and ligand molecular docking calculations. So what is target protein preparation? Which is nothing but our macromolecule. So since the raw structure has deposited into the PDB, if the structures won't be available, we have to take structures from the Uniprot database and we have to model the structure as we did it in the last video. So the model structure is like processing structure. If we are taking the raw structure from the PDB database or whatever the database, so we have to do several pre-processing steps. One is removal of water molecules, then ionic molecules, then any other conformations that may interfere with the docking calculations. So these are the extra additions which is also attached with the raw form of the proteins. So that will highly disrupt the process of binding of ligands. So then the second one is addition of hydrogens. During the crystallization process, there will be many breakage and misstructure formed in terms of hydrogens very soon. So we have to add appropriate hydrogen molecules wherever it is necessary, and we have to adjust the pH of the proteins accordingly. So these are the main pre-processing steps we have to taking care during the process of protein processing, which is also called as target protein preparation. Then active site definition. So what is mean by active site? Here you can see, let us assume this is the protein molecule and here you can see the cavity. So this cavity is nothing but where your small molecule will go and where you want to that particular ligand go and bind with the target molecules. That we can call it as cavity and binding site location are this thing. So how to identify the cavity? There are several software such as sitemap, Cascade. So these are the softwares which helps to identify the cavity based on hydrophobic and hydrogen acceptor donor. So there are several terms are available to identify the active site of the any proteins. In addition to that, if the mechanism of the proteins are already known, let us say one particular protein is interacting with another protein on a particular amino acid residue, let's say 235 amino acid residue. So around that amino acids, we can form the cavity and we can tell our small molecule to go and bind with the particular binding site or particular amino acid residue to block the particular interaction. So protein-protein interactions can be disturbed, or if any of the proteins get activated through some of the substrate binding, so that particular site also we can take it as a at binding site location. So where we can instruct our ligand, we can guide our ligand to go and bind with the particular active site, particular binding site location. So that is very much crucial step in the molecular docking calculation. So define grid parameter. Grid parameter is nothing but a box shaped structure. So here you can see the cavity. But when it comes to the actual docking process, usually they will create a cubic box. A cubic structure will be created inside the protein or around the particular amino acid to perform the affinity calculation. So that particular inside the box only, wherever the whatever the amino acid present, your ligand will go on bind with the particular amino acid which is present only inside the particular grid box. So once everything done, will be move on to the ligand preparation. So ligand preparation is nothing but to make the ligand into the appropriate form and to make ligand into the optimized structure. So these are the two important things we have to taking care during the ligand preparation steps. So first one, where we can get the ligand, that I have made a separate video previously. So there are several databases available where you can get the actual ligand and where you can get the actual ligand molecule directly. There are several databases that will give SPF, let's say PubChem, then RedBank database, which will give you the mol file, PDB file, and so on. And we have to arrange the coordinates of the ligand. If, for example, in some databases, will provide the 2D ligands, so that we have to convert it into the 3D form. Then energy minimization process. We have to optimize the ligands and some of the metal compounds and the as usual ligands geometry has to be optimized to make appropriate conformations for the docking simulation. These are the pre-processing steps which is available for the ligand preparation. Then software selection. So based on the availability and based on the experiment, we can select the software. The very first one, Autodoc and Autodoc Wayne. So these two are the open source softwares. So Autodoc, where we can perform individual protein ligand calculation. So one protein with one ligand we can do at a time. But in the case of Autodoc Vina, we can perform multiple ligand docking with the help of Python code or any other open source code. So this will be fast and standard docking protocols till today. And these are some of the softwares are mentioned, such as Glide from the Schrodinger packages, then Gold, then MOE docking. So these are some paid softwares. It may give higher accurate than the free source and open source software. So all these things will be useful for the document score calculation. So once the docking performance has completed, what are the things we will get or what are the things we need to monitor during the docking calculation? This first one is ligand sampling. So each ligand has different conformational. So because of the rotational bonds, so how many numbers of rotational bonds are available and what are the different conformations that ligand has undergone. So everything we need to consider during the docking calculation. And after the docking, we need to get at least 10 to different poses. So different poses we can get it. So out of which conformations of the ligand will get more binding efficacy to the particular binding site location that we need to identify. Then conformational steps. The flexible ligand optimization will give the conformational step. So flexible ligand is nothing but so based on the adaptability of the particular binding site location, where our ligand will undergo several conformational changes due to the weakened single bond or rotatable bond. So that conformational steps we need to undergo during the docking calculation. So once the docking has completed, we will be getting the binding affinity between protein and ligand. So what are the things we will get after this binding effect? So different softwares will provide different scoring calculations. For example, if we are performing autograph renum, we will get binding energy, binding of an score, which is nothing but kilocolor therapy, and glide XCG score. So these are all different scores are available for different websites and software. While during the ritual screening process, we may not reliable on a single source of calculation. We have to perform in multiple software or multiple scoring functions we have to use to make our calculations appropriate and the highest. So once we perform the calculations, we have to identify the interaction analysis. So interaction analysis are nothing but how these ligand and proteins are connected together. So there are different types of bonds are available, such as hydrogen bond, hydrophobic contact, and pi cation and pi ion. And these are the several types of interactions which is happening in between protein and ligand. Usually the hydrogen bonds will be considered as the most important one since it is stabilized the complex structure between protein and ligand. And hydrophobic contact also important in some few of the cases. Case to case, it will be varied and we can visualize the complex structure in several. We say five now and match from the Schrodinger packages, and these are all several packages to visualize the complex structure and how to validate. So the important aspect of docking is the validation strategies. How we can validate crystal structure comparison. So crystal structure comparison is nothing but the structure while submitting into the PDB database. Some of the researchers will submit the co-crystallized form, which is nothing but a protein which is already attached with the respective drug molecules or ligand. So that particular structure we have to take it, and we have the docking complex structure. So these two structures we have to combine, we have to map it based on the orientation of the structure, and we can easily identify the RMSD value, which is nothing but root means square deviation. So if we are superimposing the co-crystallization structure and our docking complex, so how much the deviation has happened, how much the ligand has moved from one place to another place, that we can have to identify. So as much as the RMSD value is less, we can tell that our document has perfectly processed. So based on the RMSD, while superimposing the co-crystallization structure and the document complex, we can easily validate our document studies. Then the second one is redocking. So we have to, whatever the software, whatever the platform we are using, we can redock. Initially, the co-crystallized ligand will be in the form of protein and ligand. So we have to separate it. Ligand we have to separate it and protein we have to separate it. Then with the help of our docking tool, we have to frame the grid of where the ligand was previously is present. So once we frame the grid box, we have to attach our ligand to the particular protein binding site. With the help of whatever the software we are using, whatever the website we are using, that help us, we have to do the same binding process again. So after the completion, again we have to do the superimposition of our docked complex with the native ligand. So if the RMSD is less, usually less than 2 angstrom, while superimposing will be better. So like this, we can validate our dropping strategy. Then another one is a time consuming and most promising strategy is the molecular dynamic calculation. The separate video will come for molecular dynamic simulation. So this is nothing but in the dynamic environment, as like our human body, so how a protein and ligands are interacting together, how this formed a complex structure in the dynamic environment, how much duration this complex can be stable. So if we are in the movement, so the protein ligand, how these complex are in stable state. So that we can identify with the help of dynamic simulation process. That is, we can, to be frank, we can easily say that the molecular docking calculations are like photo. So during the video time, if you are taking photo, what is the current situation at that particular time frame? That we can get it from the molecular docking. But in the case of dynamic simulation, it is like a video. So the dynamic simulation can be performed in the terms of nanoseconds, or 10 nanoseconds, or 20 nanoseconds. How these complex are behaving inside the fluidic environment, that we can identify with the help of molecular dynamics simulation. Yeah. Then, what are the applications? Visual screening, we can easily find out the potential lead compound. We can validate through the biochemical assays and binding affinity calculations, and then in for visualization we can see. So now I'm showing you a simple software to perform molecular docking calculation. For example, one of the tool named CB docs. So there are two machines are available, CP doc 2. There are many softwares available to perform the molecular docking calculation. So this is one of the field. Then another one is SysDoc. So this also is even some one click document. These are the several softwares or free websites where we can easily perform the molecular docking calculation. So here we can draw the smiles and here we can upload the target protein XYZ coordinate of the file and we can click docking. And in SysDoc also we can provide the ligand smiles and or we can provide molecule file of the particular ligand or you can sketch also. That is up to the availability of the ligands. And then second one is submitting the target. So target is nothing but the protein or macromolecule. So how we can, if you know the PDB ID, let's say 5HIE, we can simply type the PDB ID and it may ask what are the chains are available and what is the exact chain you have to select and those things it will be. And if you want to include any items or unuploading, that also you can include. Now you can upload the file also. I am uploading the sample file here, a chain, and I am using that method. So we are preparing a target protein. So here you can see, so this is our protein molecule, and this will be our breadcrumb. Let us say I want my ligands to bind here, this particular location. So here you can see box center. So I can adjust. You can see that box is slowly moving here and there. So this is x direction, then the direction of y, so then okay, then the direction of z. So we have to adjust the x, y, z parameter and we have to make the appropriate binding set. So if we want to increase the size of the box, let us say I want to increase the size of the box in the x direction, you can manually do that also. And this is y direction and we can do this z direction. So we can tweak the box based on our requirement. We can increase or decrease the box size and we can move the box along the protein structure. Once we have completed the protein group formation, you can easily go for the check parameters and if you provide your email ID, so you can perform the molecular document calculation. So this also will take little time to process and I'm going to CB dump. It's the instant you will get the document score or binding affinity calculation. So you can select one subject. So as I said earlier, here there are two options. One, we have to upload our protein molecule. Then we have to upload the ligand molecule. Here you can see there are upload protein. We have only PDB format. And for ligand, this particular website will support different ligand formats. Mol2, Mol, SBF, and PDB. In one of my earlier videos, I have said that how to convert the ligand from one particular file format to the another particular format, which is nothing but the open barrel. So how we can convert from one to another one? We can easily check it and based on the requirement we can change it. Let us go to the RCSB, which is nothing but protein data bank. Let us take one structure, 3WB8Q. So I'm just taking one random structure. So this is MAPK1, MAPK1. So I'm just downloading legacy PDB format. Here we have the file. So whenever we are downloading, we have to make the file format accordingly. So otherwise it will be deleted. So here is the file. I'm just renaming protein. So we can visualize also before going for any drawing application. We have to visualize and check whether that particular ligand has included or not. So here it is included. So we need to remove that ligand before proceeding. So I am just opening it in the notepad. We have that can be removed directly from the CB doc also. So in the protein structure, wherever the head atoms, so that also called as ligand. So here you can see the head atom is HOH, which is nothing but H2O, the water molecule that we are removing. And here also some of the head atoms are available, which is also in the name of AGS. Here we can see this is protein, and here we can see AGS, which is nothing but this particular ligand, the crystallized ligand, is already attached to the particular protein structure. So before proceeding, we need to remove it. So how we can remove it? Simply we can delete it and without space and we can save it. So this will be our processing protein structure. So now if you open to that particular ligand will go outside. There will be no ligands present in the protein. So we can upload that. If we close the other website, so here we are uploading. Yeah. So protein upload has completed. So protein has uploaded. This status will come. If any error comes, it may be wrong in the file name. Sometimes if you are downloading the same protein multiple times, so let me give up here one of the examples. So sometimes if you are downloading like this, so sometimes the file name will be like this. If you are downloading multiple times, the file name will be like this. So to avoid any such thing, you have to provide the appropriate name. So in the bioinformatics research, one of the most important aspects you have to consider. So whenever you are renaming the file or whenever you are having the file or folder, you should not give any space. You have to always use the underscore or the hyphen, right? Yeah. Here it is protein, got it, and then we can upload the ligand. So where we can get the ligand drug bank. So I'm just taking sample drugs. Let us say aspirin which is small drug. Taking for the calculation of binding of the protein. Or else we can take this ligand also. Where it is here. So there is this. This particular ligand also we can download, and this also supposed to PDB format. I think we can download the PDB file from here. So download. This is in SBF format, so we can download it. So SBF format is downloaded. So I'm going to upload this SBF here. Yeah, it has completed. If you want your results to be here, you can put your email address or something. Then you can click for the search cavity. So search cavity is what it will do. Then you can see the process also. So it will locate the binding site parameters. So it will identify where the ligand can be bind. What are the possible locations where you can get enhanced accuracy? So that you can do for the search cavities. The another type of is auto blind document. When you don't have any idea about where this particular ligand will go and bind. So in that situation, we have to provide auto blind document. So that time we have to consider the whole protein inside the grid box and we have to perform the docking calculation. So the blind docking is nothing but the entire protein structure will be considered as a grid box and can be performed for the molecular docking calculation. Right? So that is nothing but blind docking. But in another case, if you know the exact location where our ligand wants to be bind, so that also can be possible with the help of binding set prediction. These are the two different types of toggles available. Yeah. So result has came. So here you can click view results. It will be detect this one we cut pointed and it is still loading. So these are the pocket IDs and these are the volumes in Armstrong cubic. So whatever the representation by the surface, so this will be the site one, which has a huge volume. And these are the coordinates we are getting and site two, site three, site two, site five. So there are different sites available. Let us say site one. So how we have to select the site? So as I told you earlier, if we know the exact location where our ligand wants to be bind, to that location we can select, or if we are don't know where to select or where to at the ligands to bind, that time we can go for the site prediction like this. So whichever the site having the largest cavity volume, which is nothing but 1240 Armstrong cubic Armstrong. That particular site we can select and we can go for the side one we have selected and we can go for this side one. Only we to select side one. Right? Side one we have selected and we have clicking for the document calculation. Here template based documents are available, which is already mentioned here. Here our document is still progressing. Okay. So for that particular site, here we've got the Vina score of minus 7.1. See, this is Vina score. So this speed of this processing with the help of Bina. So this will be our binding affinity and the cavity wall mixing that values are mentioned. And so here our ligand is binding with whatever the amino acid reflex surface. So this surface looks indicates the amino acids or the protein structure. And here you can see some of the numbers D43, Q46, 45. So this represents the amino acid. So that individual letter represents the individual amino acid. For example, D aspartic acid, so like the revaline, so something like that. So that one letter amino acid and the number of position, the amino acid position has mentioned here. For example, aspartic acid in the position of 365. So that was mentioned. Or you can see contacting residues. So these are the residues where interacting with the particular binding site. So the ligand and proteins are attached with that particular residue in the form of hydrogen one interaction. So I think inside you can visually see, and you can download also if you want to download and you want to change the representation. So like I propose the color and the style, let's say background. So is here all something looks clear. So this also can be possible. So overall, our objective is to identify the docking score minus 7.1. So docking score will be always in the form of negative, and we have to find the interaction. So what are the interaction residues? This is present inside. That's it. So at the end, we come to the conclusion of this video. Okay, we have learned how to perform molecular docking calculations and what are the mechanism behind, what are the process behind this calculation, and some of servers, websites, servers, what we have, everything we have seen here. So we'll be seeing in the next video. I hope you have understood some of the basic terms of binding affinity prediction and what are the software tools, how to perform the binding affinity, how to interpret the results of the binding affinity calculations, everything we have seen in this today's video. We'll be seeing the next video. Please like, subscribe, and share. Thank you all.